Good afternoon. My name is Linda Marban, and I am the CEO of Capricor Therapeutics. I want to thank the Alliance for Regenerative Medicine and the Meeting on the Mesa for hosting us today so that we can share with you the progress that we've made over the past year. This is a standard forward-looking statement slide. We are publicly traded, NASDAQ listed under the symbol CAPR, and all of our filings are available on the SEC website. So I like to think of this slide sort of as the table of contents of what we're going to be discussing today. We're going to be talking about the two major product platforms that we have in our pipeline, our cell therapy product made from an allogeneic cell called CAP1002, and our very exciting new exosome platform development program, which is going to be featuring engineered exosomes as well as exosomes made by our cells, the CDCs. We have a strong balance sheet at this time with around $36 million in the bank. We have a long list of collaborators. Most notably, we've announced a partnership with Johns Hopkins University and the laboratory of Dr. Stephen Gould, where we are working on the development of the exosome platforms. In our pipeline slide, we tell this new story. As you can see, 2/3 of the products that we are developing are based on our exosome technology. 2 of the products are based on engineered exosomes, both of which are going to be deployed as Generation 2 vaccines for COVID-19, but will also be standalone products in terms of their opportunities for development of engineered exosomes. Our legacy products are our CAP1002 allogeneic CDCs, both being used in late-stage clinical development. One is for Duchenne muscular dystrophy. The other is for the treatment of the cytokine storm associated with COVID-19. And now stay tuned for the story. So what are exosomes? Exosomes are a new class of therapeutics. We have developed them ourselves over the past 5 years, but the field of biotechnology has been exploding with opportunities for exosomes really for almost the last 20 years, when we first thought they were trash cans of the cells and left them on the shelf, all the way to now, where we realize that we can finally deliver contents to the nucleus of the cell using how cells communicate with one another. These are called the exosomes. Exosomes are lipid bilayer nanoparticles. What that means is that they are basically little tiny cells, and they carry information that can change transcription, translation, and post-translational modification of proteins, which ultimately define cellular activities. They can be bioengineered, and they can be lyophilized, so they have a tremendous utility as a therapeutic, sort of the generation next of cellular or regenerative medicine, if you will. So what are we going to be using these exosomes for? Well, as I mentioned, we discovered our exosomes as we were looking for the mechanism of action of our cells, and I'll be talking about that in the next few minutes. But when we did, we realized that the information that the exosomes were carrying to derive the benefit of CAP1002 was really sort of almost a passive bystander approach. We were able to take the cells, isolate out the exosomes, and let the exosomes do what the cells were already doing. But what we realized is that we had this opportunity to take the exosomes and put in them what we want so that we can then turn them into opportunities for therapeutic development for all types of indications, whether it be for infectious disease, our first targeted opportunity with going for vaccines for COVID-19, whether it be oncology indications, we can make oncology-based vaccines or use them to deliver therapeutics in a very specific and targeted way. Exosomes are very familiar to cells, and so we can tell them where to go, and they tend to get there and derive their biologic benefit with using smaller doses than you would have to use, for instance, with a liposome or some other method of delivery. I'm very excited about the opportunity of using exosomes to deliver gene therapies. So monogenic diseases such as Duchenne muscular dystrophy, a space that we've been working in for a while. What you are currently using as a field right now is taking something like a viral vector and trying to get the gene to be inserted into the patient's genome so that we have sort of forever repair of a genetic mutation. But there's also a lot of safety risks with that. What we're able to do with the exosomes is make sort of a bridge to gene therapy, where we're able to take mRNA, put them in the exosomes, lead to relatively long-term protein expression, maybe several months, And then we can always deliver more, very safe, very easy. You're not putting a viral vector in. And so you're able to attack all types of monogenic diseases with this very simple, call it quarterly infusion, if you will, maybe more often, maybe less often, depending upon the disease, but to have a fix so that you have protein expression without safety risks. And then finally, sort of in the same guise I was just speaking of, you can use these mRNA or microRNAs or siRNAs to address all types of neuromuscular and inflammatory diseases, because what we know about exosomes is that they are profoundly immunomodulatory. And you'll hear a little bit about that through our cell therapy program. So let's talk about the vaccine program. So why is Capricor developing vaccines for COVID-19 when the field is full of those that are trying to develop them? Well, because we think that we are building a better mousetrap. We are not reinventing the wheel. We are actually building a better vaccine, or what we believe to be a better vaccine. The concept is that we're using exosomes to base these vaccines on. What does that mean? What that means is that we're using a human-derived system, the exosomes, how cells talk to each other, to actually deliver the vaccine. We believe this will be safer than using liposomes, which is being used by others in the space, and also more efficient, because you're not just shooting lots of targeted information into the body and hopefully it to get somewhere. What we're doing is directly targeting it to human cells, where we know that we should be able to derive both a antibody or B cell or T cell or cellular response, both of which are very important for immediate as well as long-term memory. So let me drill into each of these vaccine programs and talk to you a little bit about it. The first one is this one, the virus-like particle. We take the exosome, and basically an exosome is a virus without the material to incorporate into a cell and be pathogenic by making more of themselves. So you can trick the cell into believing that the exosome-based vaccine is actually the virus. And so what we do here is we take the 4 major viral antigens, the N for nucleocapsid, S for spike, M for membrane, and E for envelope. We decorate the outside of the virus-like particle with these specifically targeted proteins, and then we purify them and we can use them in order to build long-lasting immunity to SARS-CoV-2. Now, this type of vaccine is commonly used. It's used, for instance, for HPV. The reason that it hasn't become more popular, because it is a gold standard of immunization, is because it's been hard to get a human-derived system. We, in fact, now have that with the exosomes and are excited to see how these perform in human beings. The other program is our mRNA-based vaccine. This is, again, just a way of looking at the mousetrap a little bit differently. Instead of using a liposome, which is a man-made, manufactured sort of fat ball, we are taking exosomes, which are made by cells, how cells communicate. And again, we're using those same 4 viral proteins to decorate the outside, to put inside of the vaccine. So this is inside the exosome, not on the outside like the VLP. This is inside. And we deliver this, and then we believe that we will generate immunity to all 4 viral proteins again, the M, the N, the E, and the S. This one is in later stage preclinical development. We expect to come into the clinic with this program in a very short time. And please look for manuscript as well as publication soon for this particular program, which we believe could potentially be the best vaccine candidate under development. So in terms of the timelines, what are you watching for this vaccine development timeline? Well, they are both an animal study right now. We're working with small animals, and we're looking to enter into the clinic hopefully by the end of 2020, beginning of 2021, with either or both of these programs, depending upon how they perform in the late stage preclinical development. As I mentioned, watch for news on that coming soon. So now we're going to shift gears and we're going to talk about CAP-1002. CAP-1002 is our cell-based product. It's an allogeneic, it means it's off the cell. It's based on a cardiosphere-derived cell. For those of you that have been attending this meeting for a while, know that I've been presenting at this meeting about CAP-1002 for a while. We know a lot about CAP-1002. We've been working on it for more than a decade. It's not a stem cell. It's a cell that technically is a progenitor cell, but actually what we use it for is a delivery vehicle for the exosomes. So what we learned over time is that the biologic impact of the cells lasted far longer than one would imagine either a cell that's release factors for parakines like cytokines or proteins. We knew that they were not engrafting because they weren't stem cells. So how the heck were we seeing results many months later? And what we were able to do is discern that the mechanism of action of the exosomes. The exosomes contain very specific microRNAs, non-coding RNAs and proteins. We've quantified and qualified these. We know that the exosomes are then taken up by target cells, primarily the macrophage, and they stimulate long-lasting changes in cellular behavior. The cells have been in over 200 people to date and has a very strong safety profile. So let me talk a little bit about how we're deploying our cells. As I mentioned at the beginning, we're deploying them in 2 programs. One is in our COVID-19 program, working on cytokine storm associated with COVID-19. We did an emergency use authorization program, treated 7 patients, recognized the sweet spot of patients were those that were in severe phases of COVID before they got too critical. Sometimes patients are just too far gone to help. But we believe that if we intervene soon, then we can ultimately make a difference. Based on that, we have an IND for a phase 2 clinical trial called INSPIRE to treat up to 60 patients. And we expect to start enrolling that trial very shortly with patients that have severe but not critical SARS-CoV-2. And let me just highlight that here. This sort of is the pathogenesis of COVID-19. Most people get the virus, they recover, they might be miserable for a while, but they go on to live their lives normally. A subset of patients are not so lucky. They develop this hyperimmune inflammatory process. We actually, as a field, are learning more about that. Some people are just more prone to that. And what happens is these are the people that present in the hospital. They have trouble breathing. They have ARDS-induced pneumonia. And these are the patients that we believe that we can help the most. The mechanism of action of the cells drive the benefit. And shown here on this complicated but important slide is that the CDCs basically impact the macrophage. Remember I said a few minutes ago that the cells release exosomes. Exosomes are taken up by the macrophage. Macrophage are the sentinel cell of the immune system. They can either be angry and call in a lot of sort of soldiers to fight the war of the pro-inflammatory cytokines. This can actually lead to that cytokine storm. But in reality, what our exosomes do is they turn the macrophages from inflammatory to anti-inflammatory, driving back the opportunity for cytokine storm to have such draconian effects as is shown on the far right of the top part of this flow chart. On the bottom are the activities of CAP-1002. I won't elaborate on those due to the brevity of this talk, but I can say that we've done multiple clinical trials and seen similar results over time, and we're very hopeful for the patients with SARS-CoV-2A, or COVID-19 as it's commonly called. So finally, last but very much not least, is our CAP-1002 program for Duchenne muscular dystrophy. I think most of you are aware that muscular dystrophy is when you don't have dystrophin. Dystrophin is the largest protein in the body, and it protects cells from damage. The flip side of the coin of Duchenne muscular dystrophy is inflammation, and CAP-1002 addresses both the inflammatory as well as the muscle breakdown. We are targeting those patients that have lost ambulation. This is the later stage of the disease process. Those patients are sort of like the patients that medical care has left behind. There's really nothing left for them. We know that human privacy and independence is maintained by maintenance of upper limb. So we are focusing hard on those patients that have lost upper limb function and also have the cardiac ramifications of the disease, the later stage and most needy of the Duchenne patients. The phase 2 clinical trial is completed. We conducted it at 9 sites across the United States. We ended up with 20 subjects that were treated. It was randomized, double-blind, placebo-controlled. 80% of the patients were non-ambulant, and all of them were on standard of care corticosteroids, and they were 14 years of age. Because most of them were non-ambulant, we use this performance of the upper limb, a measure that is quantified and qualified and approved by FDA as our primary efficacy endpoint to be able to evaluate the skeletal muscle function of these patients. And what we found was absolutely fantastic from the perspective of the patients. We saw a 2.4 improvement in the patients that were treated with CAP-1002 versus the placebo. The patients got 4 infusions quarterly over the course of a year. This is the 12-month data, final data. And if the data is reflective of long-term benefit, you could possibly be in delaying disease 1, maybe even 2 years. And individual patients' data shown on the right in the scatter plot showing the biologic benefits to these patients receiving CAP-1002. In this busy graphic, but very important, is the cardiac function. There is nothing out there for patients with Duchenne muscular dystrophy in terms of their cardiomyopathy, even though it's the #1 cause of death in these patients. The most important figure is this one, which shows ejection fraction, or how the body needs are met by the heart. If this data again holds up with a p-value of 0.004 long term, we could be delaying the progress of the cardiomyopathy in these patients long term, and this will ultimately sustain life in terms of quantity as well as quality. The rest of this data is supportive, the most important being a massive decline in the amount of CKMB, or an enzyme that is released when cardiac muscle specifically is broken down, suggesting that the ejection fraction change is reflecting of less damage on the heart. Finally, I'll conclude today with our milestones. We have a lot coming up this year. It's a busy year for us, both with our exosome platform technology, with the vaccines, and mRNA therapeutics coming behind that, using this plug and play type of system that we've built, the INSPIRE clinical trial for COVID-19, and updates on our Duchenne muscular dystrophy program as we continue our conversations with FDA, basing the data on whether we have more clinical work to do, or whether we'll be able to proceed with accelerated approval of this product and what that will take. And with that, I'll conclude. Thank you very much for your time. We look forward to providing you with updates in the future. Thank you.